Good afternoon. My name is Ellie McLinden, and today I'll be presenting a critical analysis of a journal article on early onset dementia from the clinical neuroscience pillar. The article I have chosen defines the bounds of genotype-phenotype sequencing to explore the underlying causes of early onset dementia. This area of study is important as early onset dementia can be challenging to diagnose due to the symptom overlap with other neurodegenerative diseases, thus highlighting the importance of genetic testing for the accurate diagnosis of early onset dementia. Early onset dementia is defined as dementia occurring in individuals aged younger than 65 years of age. It is a relatively common disease, however often misdiagnosed. This is due to a more varied differential diagnosis of early onset dementia compared to late onset dementia. Misdiagnosis can be due to the clinical phenotypic heterogeneity, whereby different genetic mutations produce the same or similar phenotypes in patients. Contrastingly, misdiagnosis can also be due to the exact genetic mutations in individuals presenting themselves in a variety of different clinical presentations. Furthermore, another reason for challenges with misdiagnosis is that early onset dementia may manifest via other symptoms other than just memory loss and familial genetic inheritance. Age of onset may also vary across patients. Regardless of the difficulties to accurately diagnose early onset dementia, it is important that it is to ensure appropriate treatment course and management of the disease. Appropriate diagnosis involves the comprehensive assessment of extensive familial history, mental status examinations, neurological examinations, and appropriate neuroimaging. Before beginning my review of the chosen article, I will briefly touch on the neurological foundations and etiology of early onset dementia. Early onset dementia is most likely to run in families and be inherited via the Mendelian fashion, which is the ability of parents to pass on genes and traits to their children. There are 3 different gene mutations that are associated with the development of early onset dementia. These are the amyloid precursor protein on chromosome 21, the presenilin 1 on chromosome 14, and the presenilin 2 on chromosome 1. These gene mutations create harmful proteins that are associated with the development of early onset dementia. This process involves the breakdown of amyloid precursor proteins and damage to nerve cells. Beta amyloid proteins build up and cause plaques, and twisted fibers of tau create tangles. Plaques and tangles first attack the memory areas of the brain, but as the disease progresses, they spread to affect more areas of the brain, resulting in complete cell death. Hypothesis, aim, and rationale. The research hypothesized that the use of whole genome sequencing, WES, could be used to identify genotype-phenotype correlations in individuals with early onset dementia. This would ultimately aim to clarify the genetic clinical diagnosis of an individual with early onset dementia. And there is a significant demand for this area of research, as there is a need to alleviate problems associated with the misdiagnosis of early onset dementia. Method. A total of 38 participants took part in the early onset dementia study. Of the completers, 47% identified their gender as male and 53% identified as women. The mean age of onset was 51.7, with a range between 27 and 64 years of age. 28.9% of the patients had a family history of dementia. Sampling procedure. Participants were recruited via voluntary participation from the neurology clinics of Ruzhen Hospital and Mental Health Care Centre in Shanghai. All patients were assessed by neurological specialists, and 10 participants were identified with probable Alzheimer's disease. 9 patients fulfilled the criteria for dementia, but unknown to whether they had Alzheimer's disease or frontotemporal dementia, and the remaining 19 participants fulfilled the criteria for frontotemporal disease spectrum. Method. The research was considered by the Ethics Committee of the Ruzhen Hospital and Mental Health Centre, Shanghai Jiao Tong University in China. The committee was satisfied that the research met the requirements of the ethical conduct in human research and thus was granted approval. Written informed consent was also granted from all participants. All participants partook in neuropsychological assessments. 24 participants were administered mini mental state examinations. 7 of these patients required further testing involving the auditory verbal learning test, the Taylor complex figure test, the Stroop color word test, trail making test, clock drawing task, and Boston naming test. 14 participants who presented with psychiatric symptoms were administered the Wechsler Adult Intelligence Scale and the Wechsler Memory Scale, the Contingent and Negative Variation, and the ISANC Personality Questionnaires. Brain imaging and polymorphisms of a polyprotein E were administered on all participants, and so were blood tests to exclude acquired cases of dementia. Genetic analysis was performed via DNA extraction from participants' venous blood sample using appropriate health protocols. And whole exome sequencing was utilised to analyse the single nucleotide insertions and deletions of the 89 dementia-related genes. Results. The research identified 6 gene mutations and another 11 variants with unknown significance across the sample. Among the study sample, 68.4% of participants' initial symptoms were hypoamnesia, 71.1% of patients developed behavioural change, and 28.9% of patients developed language defects through the progression of the disease. No single participant presented with identical phenotype onset or gene mutation subtype. This made it very difficult to identify any new genetic trends. A brief overview of the phenotypes and gene mutation of each participant within each subtype of dementia was outlined. The information from WES allowed for the appropriate genetic diagnosis of 5 participants with early onset dementia. Furthermore, the research further solidified the relationship between the amyloid precursor protein, APP, presenilin-1 and presenilin-2, and the presentation of early onset dementia. As you can see on the image on the right, it represents 5 different cases in the current study. As you can see, no single case presents the same across the MRI, pathogenetic, or pedigree variables, demonstrating the difficulty associated with the diagnosis of early onset dementia. To conclude, the aim of the study was to utilise whole exome sequencing to confirm the genetic origins of early onset dementia in a sample of probable or already diagnosed dementia patients. The research was able to efficiently diagnose 5 patients with early onset dementia, thus demonstrating that whole exome sequencing is an appropriate method to use to identify genetic sequencing of neurodegenerative disorders. The rationale behind the use of WES was to identify the genetic origins of early onset dementia to avoid the classical issues involved with the misdiagnosis of early onset due to issues associated with clinical, phenotypic, heterogeneity, and exact genetic mutations presenting differently across patients. Ultimately, removing the issues associated with misdiagnosis will allow for more efficient diagnosis of early onset dementia, allowing patients and their families to receive appropriate treatment quicker, manage symptoms more effectively, and enable them to be prepared for the manifestation of the disease in the future. Strengths and weaknesses. While the present research had the ability to accurately diagnose 5 patients, there were multiple weaknesses identified in the methodological design that must be overturned in future research before the findings from this study can be legitimised. Strengths. The study utilised a combination of surveys and questionnaires to gather a wealth of information surrounding cognitive ability of the participants. This is a strength in the research because it is a simple yet cost-effective way of collecting a lot of data quickly and efficiently. Furthermore, data from surveys and questionnaires are simple to analyse, meaning that results can be more easily interpreted. Furthermore, the research was quantitative, therefore can be easily replicated and repeated in future research. This is an important strength as research is always evolving and researchers are always looking to build on their current findings. Therefore, an easy to replicate study that is quantitative is often favourable over qualitative research. Weaknesses. A small sample size of 38 participants was a significant drawback of this study. A small sample size reduces the ability to generalise results to the greater public and reduces the significance of the study findings. Future research should aim for a larger sample size. The use of MRI in the study method is an expensive and timely timely to administer. While the information gathered via medical imaging is useful and greatly informative, it is questioned whether it is needed in this study when the key aims are based around genetic testing. Lastly, the aim of the study was to set out to utilise whole exome sequencing to identify genetic origins of early onset dementia. Of the sample, it was only able to accurately identify 5 cases of early onset dementia. Thus, we are forced to question whether the limitations of the next generation sequencing technology may fail to accurately explore the genetic cause of early onset dementia. Implications. This study has demonstrated that whole exome sequencing is an effective tool able to diagnose the genetic origin of early onset dementia in some cases. However, more work must be done to improve the effectiveness of next generation genetic technology to enable greater populations of participants to be diagnosed to benefit from the development in technology. While this may not be cost effective, the findings could be far-reaching for the diagnosis of neurodegenerative disease. While this study saw some promising results, this area of research still has a long way to go. Firstly, future research could focus on addressing the drawbacks of the study presented today. Any developments in the genetic diagnosis of early onset dementia will enable better outcomes for the individuals and families affected by this unpleasant neurodegenerative disease. Thank you.